This is a PET and a CT scan superimposed on each other of a mouse that's infected with a bacteria called E. coli. And what you can see is this new probe that we have developed is lighting up the infection on one of the thighs. On the other thigh, there are dead bacteria, and they're not lighting up. You can also see some signal from bacteria, live bacteria, in the intestine in the middle of the panel, and then you can see some signal from the bladder. Now we're zooming into the infection of the thigh, and you can see these bacteria, and you can see this energy, which is what the PET imaging system captures, which is coming from the bacteria. And the energy is coming from the bacteria because the bacteria love this new tracer that we have developed, which is now accumulated at high concentrations inside them. And now this tracer is emitting energy, and therefore the PET scanner can now localize where the bacterial infection is. This image shows the new method, the bacteria-specific method that we have developed, and a direct comparison to the traditional non-specific method that's sometimes used to detect infections. So what you can see very clearly here is the yellow arrow is pointing to the real infection, and the red arrow is pointing to the dead bacteria, which is not an infection but induces what we call as inflammation. So the new method can reliably look and locate the actual infection, and you can see that very clearly with the yellow arrow. And the dead bacteria do not light up with this new method. But the old method is non-specific, and it cannot distinguish the live bacteria, which is on with the yellow arrow, from the dead bacteria, which are causing inflammation, but then no live bacteria there, which is the red arrow on the other side. One of the important things in infectious diseases is to understand if the antibiotic that you're using is actually killing the bug or not. And in this panel, you see a bug that is susceptible to an antibiotic that we commonly use to treat these infections. And on the left-hand side panel, you can see this mouse with the infection on the thigh before the antibiotic is given, and you can see very nice signal. 24 hours after treatment with this antibiotic, because the bug is susceptible, the bug is dead, and you can't see the signal anymore. Now compare this to the next panel, where we have an infection with a resistant bacteria. So before treatment, you can actually see a very nice signal, but we gave the mouse 24 hours of treatment, and actually the signal has gotten even worse. And that means the bacteria are actually not dying, and they're multiplying. And so this is very important because this allows you to provide rapid information about whether your antibiotic is killing the bug or not. And this information could be very useful in management of patients, as traditional methods can sometimes take many days to give the same information.